So now that we understand the mode of DNA replication, which is of course semi-conservative based off of the descriptions that we had in our previous video, we can now look at what semi-conservative replication really is. And we're going to entitle this next flowchart DNA Replication 2. So DNA Replication 2. And we're going to subtitle this flowchart Semi-Conservative Replication, because that's what we're going to be looking at now, because we know that that is the correct proposed model of DNA replication based off of the Watson and Crick model itself. Semi-conservative replication, and there are two parts to semi-conservative replication that we're going to be looking at in the next two videos. The first part is called initiation, and that's what this entire flowchart from this point forward will be devoted to. So, initiation. You all know that initiation means to start something. How are we going to start the semi-conservative DNA replication process? So in order to do this initiation, you're going to have the following event occur. Initiation is all about the following. It's all about unwinding of two DNA parent strands. Just like we stated before, remember, we have to take this parent strand that we have a very basic drawing of and split it up, right? We have to split it up so that each half of it can serve as a template for that new blue strand that's going to develop. And in order for that splitting up to happen, there's going to be many different initiating processes. Those processes are usually going to be done through the following mechanisms that we're going to explain. So in order to unwind this DNA, because we have to remember it's not this basic ladder structure, it's a very complicated helical structure that has all these loops and turns and complicated structure that's very, very strong, that we have to figure out how to split open, how to unwind. And in order to do that, we're going to be utilizing the following. First and foremost, initiation, this unwinding, the unwound portion. In order to unwind, we have to do this by proteins. So DNA is unwound by proteins. Specifically, there are three proteins that you have to understand and know about. The first protein, one of the most famous, there's a classic bio joke about this, are DNA helicases. Unzipping genes, literally what this means. DNA helicases, their job is to unzip genes. But more specifically, we're going to be a little more scientific by stating that DNA helicases are, if you see ASE, you know that it's going to be a protein, and thus it's also going to be an enzyme. Those are synonymous. Enzymes are proteins. So this protein, this enzyme, is going to be an enzyme, ENZ, that breaks, and you should understand this, if it's going to break the bonds right in the middle of DNA, if I told you that, what bonds will it be breaking? Covalent bonds, phosphodiester bonds? No, of course not. This is going to be an enzyme that breaks H bonds. It breaks H bonds. Do not forget that. Helicases break H bonds between where? What specific structures? The nitrogenous bases. We'll say NBs. The nitrogenous bases are connected via H bonds. Remember how C is combined with G with 3 H bonds, and then A combines with T with 2 H bonds. Helicase is going to break those bonds, and in order to do that, we're going to utilize its enzymatic quality. Once that has happened, we get the following results. The two strands are going to be separated. They're going to separate because we're unwinding, we're unzipping the genes right at the middle. You have to imagine that this unwinding is happening, oops, that's not at the middle, somewhere like this. We're having an unwinding event occur. These two strands will separate, and the helix itself unwinds as the separation is happening, because those two things go hand in hand. If you're separating strands, you're unwinding the helix. And also, interestingly enough, DNA helicase is actually an enzyme that requires ATP. It needs ATP to work. So this is not a, let's say, energetically favorable thing to happen. You need to input some energy. This is an endergonic process that needs ATP to unwind. So understand that DNA helicases don't just work on their own. They need ATP to drive them. Imagine that they're a tool, and this tool needs gas of some sort of energy, and it uses the ATP as its energy to do this very fancy breaking of H bonds right at the middle to unzip genes. to unzip DNA. DNA helicase does that. So again, our overall goal is to unwind the two parent strands. It seems like DNA helicases do a pretty good job of this. They are proteins and thus they are also enzymes. In addition, there are some nuances. There are some things you have to understand about unwinding, some consequences, let's say. The number 2 protein that you should understand are single-stranded binding proteins. Single-strand binding proteins. These are very interesting and very, very important. They're more often just abbreviated as SSB proteins, single-strand binding proteins. What they're going to do is you have to imagine the following situation. DNA is going to be unwinding, right? So DNA unwinds. Big deal, right? DNA unwinds. So what? Dot, 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 right? But what's going to happen is you need to stop it from rewinding back on itself, from going back onto its regular structure. Need to stop it from winding back, together specifically. Because DNA, naturally, because those H bonds are so favorable and DNA has this beautiful, beautiful structure that's so favorable in nature, it will tend to go back on itself if it has the opportunity. But right now, you want replication. You want to unwind. You want to make sure that they stay unwound. So in order for this to stay unwinded, or unwound, I don't even know which one it is, you have to throw on some SSBs. And specifically, these SSB will bind to single strand, because remember, once we've split them up like this, what I'm going to do is I'm going to take some SSBs, these circles, and attach them right there, and attach them right there, so that these guys stay away from each other. SSB will bind to single strand to prevent, their preventive mechanism, their preventive protein to prevent reformation of double helix. So that's number 2. So we have helicases, great, they unwind. How do we make sure that it stays unwound? We use single-stranding binding proteins to keep them from rewinding back on themselves. And what's our third protein? Our final protein in initiation is going to be, or are going to be, a class of proteins called topoisomerases. Topoisomerases are a class of proteins and also a class of enzymes, you see that ACE ending, that are going to aid in the following. Their job is they do the following. They break and rejoin, topoisomerase breaks and rejoins backbone molecules. So, this is a very weird function for an enzyme, right? Why would you break and then rejoin something over and over and over again as it does in DNA replication, specifically during initiation? This seems counterintuitive, but this breaking and rejoining is a mechanism that prevents, overall, it prevents knotting up of strands. Well, you have to imagine, and I think a great analogy is a rope. If you take a rope or a string like this, right? This is a very basic string or rope, and you want to split it apart right at the middle. You're going to start splitting it apart like this, right? It's going to start splitting, but as you keep on splitting it apart through the middle, as you keep on taking these ends further and further away from each other, you're going to end up with a lot of built-up pressure that's going to keep on radiating down and down and down this whole string as you try to split it apart. The same exact concept is happening in DNA, and in order to prevent the knotting up that's eventually going to happen over here, where it's going to be so thick at this end that you're not going to be able to move forward, you're not going to be able to split it apart, you're going to have to use topoisomerases. These overall, their goal, these two facts are going to allow topoisomerases to relieve, a topoisomerase relieves strain, it's the key word here, it relieves strain from unwinding. Unwinding is not a very natural state that DNA wants to be in, and in order to make sure that it stays calm and stays okay and doesn't just absolutely break down or get so wound up, no pun intended, when you're splitting apart this rope, imagine, you have to relieve strain. And in order to relieve strain, you use topoisomerases that create what is known as a relaxed conformation of DNA. Relaxed conformation is a great way to make sure that DNA stays happy during this very sort of invasive process of unwinding. In addition, the final two things that we're going to be talking about in this video are the origin of replication. I'll do that one right now over here. Origin of replication. And also we're going to be talking about replication forks. We'll do that one over here. Two final points on the initiation process. The origin of replication is very simply going to be referred to as the location, and it's exactly what its name refers to, location within DNA molecules where replication begins. Nothing fancy, nothing crazy there. The origin of replication is the location within a DNA molecule where replication begins, the origination point. More specifically, this is the first point, the first point of separation and unwinding. Two very basic facts that you should understand simply from the name. First point of separation and unwinding. And finally, it's important to understand sort of a nuance of this is that there are several origins of replications in eukaryotes versus just one in prokaryotes. And this is mainly due to the complexity. We have much more complex genes, much more complex genomes than our prokaryotic ancestors, and thus we're going to have this several origins of replication and only one origin of replication in prokaryotes. The overall consequence of an origin of replication is that it creates a fork. And that's, of course, what we're about to talk about right now over here. This fork that's created, these replication forks, are very simply defined as the point at which it's the idea that both new strands, because remember, we have to make two blue strands over here, both new strands synthesize simultaneously. Okay, so the DNA makes sure that once it's in this conformation, it does everything possible to get both of those two unwound strands completely synthesized from their new sort of blue counterparts. I'll quickly draw those blue counterparts right over here. So in order for this to happen, in order for this blue part to show up, it actually happens simultaneously on both new strands. Synthesized simultaneously off of parent template. I highly suggest going on YouTube, looking at the playlist that I have for DNA replication in order for you to see this event in 3D animation. It really helps this topic click so much better in your head as opposed to just words. I highly suggest looking at the playlist for DNA on the website, which is very easily accessible on the playlist section. And finally, we're going to close out with this final topic, final point. This is done via bidirectional. It's called bidirectional replication, which simply means bidirectional. It's not just a fancy way of saying that this replication, the replication of, let's say, strand 1 and strand 2, is actually happening in opposite directions. And we're going to see that as we move forward. Occurs in opposite directions. We're going to see what we mean by that as we move forward in DNA replication, and so we've covered initiation. Initiation is a process that involves three very important complex proteins that do very important jobs listed here. There's an origin of replication, which will lead to replication forks, which leads to bidirectional replication, which we can continue to talk about in our next video.